Have you ever wondered why you have your mother's eyes or your father's smile? The answer lies in a fascinating field of science known as genetics. Genetics is the study of heredity, the passing of traits from one generation to the next. For centuries, humans have observed these patterns of inheritance, but it wasn't until the mid-19th century that a scientist named Gregor Mendel unlocked some of its key secrets. Mendel, often hailed as the father of genetics, conducted groundbreaking experiments with pea plants. He meticulously tracked the inheritance of traits like flower color and seed shape, meticulously recording his observations. His findings laid the foundation for our understanding of how traits are passed down through generations. Before Mendel, heredity was a mystery. People knew that traits were inherited, but they didn't understand how. Mendel's work revolutionized our understanding of inheritance. His discoveries, however, remained largely unrecognized during his lifetime. It wasn't until the early 20th century that Mendel's work was rediscovered and its significance fully appreciated. This rediscovery sparked a revolution in biology, leading to the birth of modern genetics. To understand genetics, we need to familiarize ourselves with its language. Let's start with the fundamental unit of heredity, the gene. Imagine genes as tiny blueprints within our cells that carry instructions for building and maintaining our bodies. These instructions determine our traits from the color of our hair to our susceptibility to certain diseases. Genes, however, don't exist in isolation. They are located on structures called chromosomes, which are thread-like structures found within the nucleus of every cell. Humans have 23 pairs of chromosomes, inheriting one set from their mother and one set from their father. Now let's talk about alleles. Alleles are different versions of the same gene. For example, the gene for eye color has different alleles, such as those for blue eyes, brown eyes, or green eyes. We inherit two alleles for each gene, one from each parent. These alleles can interact in different ways. Some alleles are dominant, meaning that they will always express their trait if present. Others are recessive, meaning that they will only express their trait if two copies are present. Understanding these basic terms is crucial for unraveling the complexities of genetics and how traits are passed down from one generation to the next. Gregor Mendel's experiments with pea plants were instrumental in establishing the fundamental principles of inheritance. By carefully crossing pea plants with different traits, he observed patterns in the way these traits were passed down to their offspring. Through his work, Mendel unveiled two fundamental laws of inheritance, the law of segregation and the law of independent assortment. The law of segregation states that each parent carries two copies of each gene, but only one copy is passed on to their offspring. This process of separating the alleles is random, ensuring genetic diversity. The law of independent assortment, on the other hand, states that the inheritance of one gene does not influence the inheritance of another gene. For example, the gene for pea color is inherited independently of the gene for pea shape. These laws revolutionized our understanding of inheritance. They provided a framework for predicting the likelihood of offspring inheriting specific traits from their parents. Mendel's work laid the foundation for modern genetics and continues to shape our understanding of heredity today. While Mendel's work provided the framework for understanding inheritance, the molecular basis of heredity remained a mystery until the mid-20th century. In 1953, James Watson and Francis Crick, with contributions from Rosalind Franklin, made a groundbreaking discovery. They unveiled the double helix structure of DNA. DNA, short for deoxyribonucleic acid, is the molecule that carries the genetic instructions for all living organisms. Imagine DNA as a twisted ladder with the rungs of the ladder made up of four chemical bases: adenine, A, thymine, T, guanine, G, and cytosine, C. These bases pair up in a specific way. A always pairs with T and G always pairs with C. This sequence of bases forms the genetic code, which determines the instructions for building and maintaining an organism. The discovery of DNA structure revolutionized biology, providing a molecular explanation for how genetic information is stored, replicated, and passed down through generations.